a whole host of co-authors, many of whom are here. A lot of this work is funded through the USGS Status and Trends. So my Great Lakes Science Center, who we work for, is basically chartered by Congress to serve the fisheries information to the states and provinces of the Great Lakes. Also the DEC, obviously we work with them, and OMNRF and the Canada-Ontario Agreement. So quite a lot of funding sources contributing to different parts of this work. Take-home messages really from the talk. First are thinking about round goby and round goby behaviors and the impacts on them. But if I can, to take a chance, really to do a little bit of an advertisement, is to focus on the collaborative opportunity that our prey fish work provides. You heard about the Prey Fish Working Group. We're out there often studying a lot of different things, right? And you saw this picture before, but we have the big boats, the lake coverage, and we're out there seasonally. So if you think of, like, if you're interested in these interactions, there are collaborative opportunities. You can come along. We can provide samples. For instance, Dr. Legard at Brockport gets a lot of his prey fish for thiamine, thiaminase, fatty acid work from these samples. Other people, University of Vermont, a whole bunch of slimy sculpin genetic samples went out there. Because we're out there, we're happy to do that. We see that as a point and a value-added role of why we're there. But there's a lot of data too. We've been out there, as Mike said, for over 50 years. There's a lot of databases. Dr. Suresh Sethi at Cornell is using the alewife data to work on predator-prey modeling. In fact, just before I came here, I got a request from a SUNY ESF master's student to use all the lake trout diet data that we have from these tows. And so it's a resource, and there's my contact. All my other colleagues are here too. Please talk to us if you're interested. Whatever you want, we can try to find it. So really, as we think about round goby, this isn't, I'm not going to give a talk on any one specific study that we've done, but I wanted to start out with just some of the bigger picture observations about goby and their role in the system. And the first is this critical link between food web link, between dreissenids and piscivores. So you're hearing that, they're showing up in diets. And so if you had mussels without gobies, some folks have called it sort of a trophic cul-de-sac, or it's energy that's not moving up through the food web. In 2013, we went out almost 2,000 diets we collected from predators. We found very similar to what others have. Some species rely heavily on round goby. The darker bar here is the percent of goby by weight. That's what matters to the mass, not how many, the weight of goby in the diet. Well over 3/4 of the diet of perch and smallmouth bass is goby, but also brown trout, walleye, lake trout. As you heard other folks, Dr. Roseman mentioned, Chinooks did not consume them. He did not find any. But I like to talk to the anglers about this because they're usually, shaking their fist at the mussels. It's all the mussels' fault. Well, if you think about that a goby is 70% a mussel, in ultimate, your yellow perch that you're out there targeting, 50% of them comes from a dreissenid mussel. So that's just something to think about. I was glad that Eric put a Save the Gobies poster or slide up, because I used to have one that said Save the Mussels. So just different ways of thinking about it. They've certainly restructured the benthic fish community. Here's just a typical community composition, again by biomass, from our bottom trawling in October that spans from basically 6 to 225 meters. It was a slimy sculpin-dominated system with a bunch of near-shore native benthic fishes, spot tails, Johnny darters, trout perch. Since round goby have invaded, you see most notably this just absolute plummet of slimy sculpin proportionally. And round goby have come to dominate. This is just the benthic community, remember. Interestingly enough, the native deep water sculpin that Mike alluded to has come back as well. But to put this in context, right? Round goby are important, dreissenids are big, but if we think about this in a mass balance context, and we do a mass balance model for the lake, dreissenids are about 22% by biomass of all the grazers. So zooplankton still dominate the lake. It is not just a dreissenid lake. So of all the things that graze on phytoplankton, dreissenids, per unit area, lake-wide average are about 22%. Around Goby are 2% of the prey fish. Because, as Mike said, this is still an alewife lake, right? It is still heavily dominated by alewife. So while we're talking about Goby and they're kind of the new hot thing to talk about, the major prey fish biomass remains to be alewife. So in thinking about the food web role around Goby, really we need to understand two behaviors that impact that role. And the first is the diet, right? You've heard folks talking about that. I'm going to show you some of the information that we have that we've been understanding how they eat. And then also the seasonal movement. And you can imagine those interact, as Dr. Panuto said, where they're moving to, where they're eating, and how that plays in to the biogeochemical cycling in the lake. We'll start with the diet first. So my colleagues put out a paper initially on Lake Ontario, the first diet paper, showing it was mainly dreissenids. About 70% of Goby diets in the early 2000s was dreissenids. We went back out, double checked that in 2013, same thing. By weight, dreissenids made up about 70% of the diet. Something that's interesting, mysis. This is Mysis diluvianum. You're thinking, wait, the deep water opossum shrimp that vertically migrates, they made up 12% of the diet in this case, similar here, and that has to do with that seasonal migration, where they're moving. They're moving out into that habitat during the winter months and consuming what most people would not think of as a typical benthic food source. Because we had a whole lot of technicians who paid very close attention to things in 2013, we were able to figure out a little bit more about what gobies were eating, what was in that diet. As you know, a molluscivore has the pharyngeal teeth and arches and just crushes everything, making diets a real pain. But if you go in and understand that there's a little point there at the hinge that you can measure, and you often still find it in the diet, and you can understand the size of that hinge relative to the shell size, all of a sudden we can say, all right, a goby of what length eats what size mussel. First thing you'll notice, that ontogenetic shift that Dr. Panuto was talking about, somewhere there right around 60 millimeters. We never found, we had plenty of diets of gobies less than 60 millimeters, never found a dreissenid in them. Also, the size. Randy Jackson mentioned to it, 13 millimeters to 8 millimeters. We found thousands of dreissenids in diets. They're all in that range. And that there is sort of this preference for larger goby to eat larger mussels. We also work with stable isotopes as an information. You heard Eric give a nice description of the role that isotopes can play in helping you take kind of a longer term look at energy tracers and using your standard carbon to nitrogen biplot where paraphyte and phytoplankton gradients are on the x-axis and trophic position. If you measure goby, the first thing you might think, whoa, they're pretty varied. Like why are goby that eats mussels having multiple trophic positions? Are they eating predatory mussels? Like what's going on? But and it looks like they're eating across a size range or across a gradient from paraphyte to phytoplankton. But what's hard, and sometimes people want to use isotopes as this, shortcut to diets. If you don't know how to interpret everything in the community isotopically, you don't know how to interpret your isotopes either. And here I used dreissenids plus their trophic enrichment. What we see is that exact same sort of hook pattern. And what's interesting is this bears out to depth. So the isotopic value of mussels that are shallow is here, contrasted to 100 meters here. And we know this, if good ecologists will know that all the stuff we know from isotopes come from the geologists. They understood to know how to work in lake sediments, you need to know how the isotope changes as the phytoplankton sinks. Those models that predict that come out exactly this way. Nitrogen values increase as the phytoplankton sinks down through the water column and bacteria are working on it. So implications for this behavior, you heard it, it was funny that Randy said he didn't think they could control mussels. We have pretty good evidence they're controlling mussels. Here's a time series that I worked with Don Dittman on, where you see the initial explosion of dreissenid mussel density. We've extended this out a few more data points. Mussel density is declining, mussel biomass is not. I don't have that associated figure, but densities are certainly on the decline. So now let's talk about the seasonal movement and what we know from our sampling sites. Well, that's luckily because we go out there so often and trawl in what would be Goby habitat to some. On the South Shore, we don't have the rocks, but we have the Goby, so we can trawl there. We see in April, the Goby distribution is out here from 50 to 150 meters, right? So you think about, oh, it's a near-shore fish, except we're finding them out in 150 meters. In June, we go back out with the same gear, which see importantly is most of them are inside. In fact, most of them are probably on the inside of where we can trawl. That's where the methods that you're seeing with the video could become really important. There are still some out deep. This is the relative density on the y-axis. By July, the numbers are starting to slip out. The density, this is where we see the distribution by depth. But then again, by October, now they're out here around 60 meters. This pattern holds up for every year. We've looked at it really since about 2004. Deep, inshore quickly, then sliding back out slowly. But we do not think this necessarily holds for all places that round Goby move. We think they move, but maybe there's differences due to substrate or slope of the lake bottom. But why go so deep? Why like 150 meters? So we think that, remember we talked about this size range, what they're looking for. Refer to that sort of as Goldilocks size of mussels. There's plenty that are smaller, there's plenty that are bigger, but those are the ones that are just right. And if we look at our mussel size frequency coming down that bank, so here shell length is on the bottom, it's your basic frequency graph, and depth is each panel, what we see is there's just an absence of those correct size, that Goldilocks size, until we get out here to 100 meters. So we think that while gobies are moving, that migration is, they don't, while they can be inshore, lakes freeze, coldest water's at the surface, they want to find that warmer water. What's helping them to spread out along that bottom is this mussel density, and mussel density specifically within the sizes that they're looking for. So it often surprises people, but there's mussels in the very middle of the lake, 250 meters, so there's actually quite a lot at 200. So we use this, you see these distribution plots, we can just use the mean depth to calculate and describe the habitat use. Here the mean depth is the density we're catching at any one particular trawl. Maybe the mean depth there is 45. In the spring, really it's late winter, and if you know what the Great Lakes are like in April, it's more like winter. Here the mean depth is more like at 100 meters. So we're interested in what really is driving this mean depth. What's telling them when to go, what also influences where they go, and what's really interesting is Goby size is the most important predictor. No matter when we're looking at our trawls, be it April, June, July, or October, a bigger Goby, the mean length of Goby, always higher, offshore, deeper, excuse me, probably offshore to everybody, but deeper, bigger. And so that is pretty interesting. Who's out there in the greatest depths are those biggest gobies. Temperature, a little. We don't have a lot of variability. We have much more variability in things like depth and size than we do in temperature. But in 2014, the big chill, right? We didn't see gobies move in as quickly. So there's definitely a temperature signal that we can pick out of our trawl data. But we can pick out some timing. We do know in the fall, this change in mean depth, a little more leisurely, right? Here we're actually sampling across October, and we're seeing the size distribution, or excuse me, the depth distribution get deeper, maybe about a half meter per day. Whereas in the spring, it's much more fast. It's 1.5 meters per day. Now that's a change in the mean depth. That doesn't mean one Goby is doing all that, but it's a difference in the rate, right? So you can imagine that spring pulse versus a fall slow movement. The implications. If you do have a negative interaction of Goby, this movement spreads that interaction out. Mike showed you some of the stuff on slimy sculpins, but they're influencing any fish, any benthic-oriented fish, from as shallow as they can get out to 150 meters. The spring nutrient pulse is impressive. We actually estimated it in our 2012 report, and you know, just put a ballpark back of the envelope estimate on how much biomass was moving in and being deposited. And limit egg predation. Everybody wants to say gobies are going to be egg predators, but if they're not there, like in the fall, they have the potential to not be the egg predators on things that were typically fall spawning lake trout, cisco, bloater, stuff like that. So with that, I finished with this picture because while a little bit gross, it does just demonstrate that some of these fish are incredibly important to the sport fish. I picked a brown trout. If you're following what's happening in the Great Lakes, this is, we kind of made the call a couple of years ago, it's like brown trout should be the fish of the future. It's a little bit, it doesn't have to have the alewife like the Chinook salmon does. You can still make some anglers happy and they can make use of the Goby. So with that, I'll take any questions. Got 2 minutes. Yes, Rich.
Brian, your hypothesis that you suggested maybe the Goby are foraging on mussels when they go deep there, do you have guidance when they're deep? Or
absolutely.
Okay, and you see the mussels?
Full of mussels.
Okay. So at what point do you expect to see suppression of those size classes in the deep areas?
Well, I mean, I think we do already see it to some extent, right? If you think about the when and the temperatures they're at, they're certainly not cranking when they're down there 4 degrees C. They're not eating a lot, but so I'm not sure they would ever actually suppress the value. Went back and checked in the spring and the fall to look for that and didn't see a big change. The change was all, we should have sampled at 75 meters obviously to see the change, but it's somewhere right there less than 100. Yes, Ken.
So Brian, I see an interesting analogy between your fast and slow seasonal change with birds. Because they hurry to a high productive area to set up territories. And they get breeding done and then the territory breaks down and they can take their time moving south.
Interesting.
So maybe there's something.
I like it, yeah. Yes, Scott.
Brian, what's going on with your deep water sculpin you showed in the very beginning? They seem
Blowing up.
Like coincidental though with the Goby introduction, wasn't it?
It does look really strong, doesn't it? It's like, oh, here come the Goby and here come the deep water. So if you want native species, put in Goby, right? We have a paper that's out in the special issue on that right now. We don't think there's a lot of interaction there unless that, some of you probably went to school with Steve Brandt. He proposed that slimy sculpins and deep water sculpin, that interaction is what limited those two species. Goby negatively impacts slimies, it's got to help some deep waters. So, yes, quickly.
Like Goldilocks, do you really mean little Miss Riding Hood there?
Wait, did I have this analogy wrong?
It's 3 bears.
It sounds like storytelling.
Porridge is just the right temperature, is what I'm saying. You're right. All right, got that one wrong, but you picked up on it. You fixed it for me. Thank you. All right, for the last speaker in the session, Dr. John Farrell is going to talk to us about what they're seeing on the St. Lawrence. A couple quick announcements. Stay in here as long as you want. John has a lot of different slides about a lot of good things. The end will be when you're done talking with John. So, but can you, if there anybody who did any judging, can you hand me your forms? Thank you, Jim, very much. We're going to announce the winner of the Best Student Talk and Best Poster in 2 rooms down. The first room, is that E?
Yeah.
That one. Kurt Jerka is the moderator there, so we'll be talking there. So if you can, please hang out just a little bit longer after. John's in charge. See you soon.